All good. All right, I'll start off and talk about my potting mix, and then Bruce will talk about his. What I've got here is a sample of what I call my general mix. I use this on a wide range of plants. It's particularly good for most of the aroids. I do modify it for many of the anthuriums. But this is a fantastic mix I've found for the likes of philodendrons, alocasias, but I also use it on a wide range of other plants. I use it for begonias, crotons, flowering shrubs, and it's good if you're growing a wide range of plants. If you can sort of standardise your mixes a bit, it makes it easier to manage watering and various other aspects. So this is basically 70% 60-40 coir. So it's the coir blocks that have got 60% fines and 40% chips. So I'm using that 70% of the coir and 30% coarse perlite. And into that I put a controlled release fertiliser. My preferred product is 6-month planter coat, which has an NPK of 14, 4, 12. So it's a fairly high potassium fertiliser compared to many of the others. But they all work. You know, there's Osmocote, Nutricote, Fluorocote, etc. They all work. They wouldn't be on the market if they didn't work. I just personally have come to prefer the planter coat over the years. Some people prefer to use a shorter release product, maybe a 3 to 4 months product. Some people like to use a longer release product. Of course, the longer the release time for the controlled release product, the more you put into the mixture. So the recommended rate for the 6-month planter coat for fast-growing plants is 5 kilograms per cubic metre. I put in 4 kilograms per cubic metre because I do add this product as well, which is the microbial rock minerals. So this product that's here is called Garden Delight. It's manufactured in Toowoomba, and the Garden Delight has got beneficial microbes in it, and it's got the rock minerals. Now, one of the interesting components of it is the calcium. This product has 4% calcium. Now understand that the controlled release product, the planter coat, doesn't have any calcium in it. And calcium is a major element. It's one of the elements that plants need in large quantities. So if you're not getting it out of the controlled release, traditionally I used to get it out of lime or dolomite because I used to add lime and dolomite to bark-based potting mixes, and the lime or dolomite would control the acidity reactions that you get from bark-based mixes. So that worked perfectly. The issue with the coir, though, is it's already at a pH neutral, and it's very reluctant to shift its pH to the acidic zone. So calcium in the form of lime or dolomite may not be the best choice. It's a choice, but it's not necessarily the best choice. That's why I go into these beneficial microbe products. And the other thing that's in these products is silica. Like in this product here, it's 26.8% silica. Now the truth of the matter is silica is not an important element for plant growth. It is not one of the 13 elements essential for plant growth. I mean, if a plant doesn't get enough of any one of those 13 elements essential, it will show symptoms of deficiency. So the silica, though, enters the plant root system and the plant's tissue just the way the other nutrients do, but what silica does when it builds up in the plant is it strengthens the plant's tissue. It's particularly good for building up leaf tissue, strengthening leaf tissue, so much so that when fungal spores land on leaf tissue that's high in silica, it's scientifically proven that the fungal spores will germinate, but they have trouble entering the leaf, growing into the leaf and infecting the plant because the silica is protecting the plant. So the other, not only with aroids do I find this beneficial, but also particularly useful with orchids. I used to get a lot of problems with spotting on orchid foliage. You know, black spots which would be often fungal based, and since I've been putting the Garden Delight into my orchid media, I'm growing orchids with much cleaner leaves. So it does certainly make a difference. So I'll let Bruce talk about his product here now, because he's got a product that's quite a bit different to mine, but still very useful.
Alrighty, what I'm mainly growing tillandsias that don't need mix, but for my anthuriums, I'm using probably larger particle sizes than what Steve's using. Probably a third of the mix is chopped coir, so it's the actual large husk that's chopped up into sort of almost centimetre-sized pieces. I'm also using the jumbo perlite, which is a similar sort of size, much larger than Steve's. I'm also using 5 to 10 mil charcoal, and those three large particle sizes help to open up the mix and improve airfield porosity. On top of that, I also add a little bit, probably about 20% of the coir pith or the dust, and that helps to fill in between those huge particles. What I've sort of found over time is that I've been keeping, a lot of the roots on my anthuriums too wet, so I've been opening up the mix, hoping to improve the drainage and allow it to dry out between waterings. I'm sort of sure that a lot of my problems have been that I've just been growing them in too heavy a mix and keeping it too wet. And especially during the cooler months, that can be a big problem. And then obviously as your plant grows and gets bigger and gets away, having those larger particle sizes helps, just for airspace for the roots to grow between, as they get to maturity. When I'm mucking around with smaller stuff, I'll add probably a third of just your general commercial, fine potting mix to this mix, because, I'm dealing with quite small roots and small plants. One of the first questions that we had, and I use a slow release fertiliser. I prefer Osmocote. It's something that I've used as a commercial horticulturalist over the years. And like Steve, we get stuck in our little ruts with fertilisers. I always thought we did some trials way back when and we're happy with the product. Obviously, just remember that your slow release fertilisers tend to dump their nutrients when we have extreme weather. And so, when we start getting some warm days in September, October, you've just got to watch out that you're applying enough water to leach out any of that overdose that we might have. One of the first questions that we had from Sandra, she was just asking if she can get some tips on how to germinate, propagate anthurium seeds. When's the best time to do it and how, and any tips for potting them up and post-germination. And we've got this little tray as an example, and I'll tell you what I do, and then we'll look at this tray. Basically, I'm using the jumbo perlite in the bottom of my seed pan, and then I put a layer of the coir dust on top, and it's only probably about 1 centimetre to 1.5 centimetres deep of that coir dust. And of course, anthurium seed needs to be sown ripe, fresh, straight off the plant, and you need to remove the outer coating of the fruit, which is usually the coloured berry. And the best way I found to do that is to sort of give them a gentle crushing, squeezing action in a small bowl, and then add water, stir it up, and you can tip the fruit off, and usually good ripe seed sinks to the bottom, and it's kind of like panning for gold. And if you do a lot of it, like I do, you can get really good and you don't lose too much down the plug hole. But that's an issue. So basically, I sow that directly onto that coir dust with the perlite down the bottom, and within a week you'll see movement and you'll see germination. And what I do is prick them out of that tray if I need them straight away. I'll do them as soon as I possibly can, usually at about 1 or 2 leaves. And the reason that I do that is the root, what you're seeing on top with Anthurium generally, there'll be a single root that's going straight down or going sideways or whatever. And if you sow heavily into that tray, they'll start to intertwine and they'll start growing together, and you'll end up kind of something like this, which pulling them apart can take time and effort, and you may damage the roots and whatever. But if you're doing them very quickly in their growth stage cycle, you can get them where the roots are only 1 centimetre or maybe 1.5 centimetres long, and you can prick out thousands in a day very quickly and put them into cell trays. Whereas if you leave them like this, it becomes a massive task to get them apart. Now, these are sown into sphagnum. In nature, that's kind of what you'd see in the bush, but in a commercial growing system, it's too tricky to pull them apart from the moss. And the other thing about the perlite at the bottom is that A, it provides perfect drainage, but B, if you do leave them when they get to this sort of size, they're much easier to pull apart rather than if they were intertwined in amongst all the, say, even coir, they can get stuck together quite badly. But the perlite I've found over the years, just they fall apart very simply, and they're much easier to manage in numbers. And, you know, once you get to a certain, well, you know, when you're doing it commercially, you haven't got time to waste, and you don't want to be damaging your plants. So that's why I do it my way.
And I do it my way. I put some of this general potting mix in the bottom of a squat 140 or 165 mil pot, depending on how much seed I've got to plant. And then I put about 50 mil, or no more, less than that, about 30 mil of sphagnum over the top of the potting mix. But because of that problem that Bruce talked about, how separating them from regular sphagnum, I get a very long pair of very sharp scissors, and I chop the sphagnum up small. So it's really small pieces of sphagnum, and when it comes to separating the fruit from the seed, rather than that, that's a medieval practice that Bruce was talking about, squeezing. Years ago I learnt this trick. You go to Joyce, Maine, and you buy a cheap food blender. And you know those steel blades that you use to make your smoothie, that flog all the ice and the banana up? You get irrigation black microtube, and you heat that up, put that in boiling water, and then you push it over the steel blades. And then when it cools down, you chop it off at the top of the blade. And then you go and get this big infructescence of Anthurium seed, cordyline fruits, great big bunch of philodendron, chuck it in the blender, 3/4 water, zap it for 30 seconds, stop, have a look. Not quite finished, zap it for another 30 seconds. What'll happen? All the seed will go to the bottom, and all the fruit floats to the top, and then you tip it off, and the fruit all floats off, and you've got to look in there. There's still a little bit of fruit stuck on some of those seeds. Another little zap. Don't overdo it. Just get it right. And before you know it, it's all ready to plant. And the other thing to understand is that the fruit on Anthurium and philodendron seed contains a chemical inhibitor that slows the germination of the seed. That's why you must wash the seed before you plant it if you want to get it up quickly. It'll still come up if there's an inhibitor on it, but it'll take weeks instead of days. And so that blender thing, that really gives it a good clean. The only time I've had problems with it is some very large seeded species where those blades just seem to knock them around. But that's how I do it. It works.
That's good. We've got another question here. Robin would like to know how to successfully propagate new plants from the non-root subterranean aeroid parts, which some call corms.
Okay, so here we took and
We're
talking Amorphophallus.
Alocasia.
With
Caladium.
Caladiums are tubers, but the same principle applies. Yep. Tuber being a modified stem. So they're both modified stems, corms and tubers. But a caladium tuber, you can cut it up into small pieces and you can grow pretty much every piece as long as there's an eye in it. They have little eyes like a normal, English potato that we eat. With the alocasias, though, it's really a matter of separation of bulbils. We can't cut one up. You've got to really separate the individual bulbils off the main bulb.
I think, I think traditionally it's sort of like a late winter, early spring job to go through the mix in the old pot. What I've done this year on a couple is separate them out, clean them, pick out the good ones, and then just arrange them growing point up.
Yep.
And probably an inch of soil on top of that, and away they'll go.
So when you cut up the likes of a caladium structure, you need to let it dry before you plant it. Let it completely callus over where it's been cut. A practice that some people use is to where they've cut the thing to actually that cut surface to coat it with a bit of lime, and that helps to stop any bacterial or fungal infection getting into the tissue, but also helps it to dehydrate faster and actually callus over.
Alrighty, this is another one from Keith in South Australia. He'd like to know, what are the hardiest aeroids to grow for us growers who live in the southern areas of Australia? I think probably the easiest way I could explain that is you've probably got to look at plants that occur at either the northern or the southern range of where aeroids occur in their native ranges.
And high altitude stuff, probably?
Yeah, well, often the high altitude stuff, if they have like a hot day,
they won't take a heat wave.
They never get adapted to the heat. So it's more they've got to have some adaptation to cold blasts of air that would occur in their native range. So I know in southern Peru and Bolivia, they have very cold winds that blow up from the south, and they can regularly go down to 7-6 degrees, and they're still what we would consider incredibly tropical plants. Tropical looking plants, but they've got that natural adaptation. So I think, some of the Central American, Mexican species are probably better things you should be looking at, and probably Argentinian, Peruvian, southern Peru, that's...
Yes.
There's
aeroids that occur in pretty much every climate on the planet, but a lot of them are obscure and we don't sort of see them offered for sale and cultivation.
No.
But the only places on land that you won't find aeroids is inside the Arctic Circle and inside the Antarctic Circle. But there are species of aeroids that go right up into those very cold zones in the northern hemisphere. But it's just a matter of searching them out and finding them because they do exist.
They sure do. Jess has got a question. What fertiliser regime do you use coming into spring compared to winter? And what ratio of nutrients do you use? How often do you fertilise?
Well, coming into the warmer weather, that's when I continue to do exactly the same thing as I do in my potting media, but once I come into the warmer weather, that's when I might start to up the ante with a bit of half-strength foliar. Yep. Where I'm boosting the spring growth with some liquid feed in through the leaves, because the uptake from the root system won't be back to maximum or optimum until the media warms up properly, and that can take a bit longer in the pots for the roots to get warm. So yeah, a bit of foliar doesn't hurt. And there's a whole lot of products on the market. They're all really good. I use organic products, but there's a lot of different products out there, and they all work. Otherwise, the company wouldn't still be in business.
Just on the foliar fertilisers, I use during autumn and winter a product that's higher in calcium and magnesium called CalMag Finisher, and then in spring I'll change to a what they call an all-rounder, which is higher in nitrogen. So.
Well, calcium hardens plant tissue too. It's very important, and particularly not only in aeroids, but Stan would probably know that Dennis Andresen, who used to have the palm plantation up at Buderim, he would always fertilise his palms in autumn with calcium nitrate, because that form of calcium is highly soluble. It goes into the plant tissue very quickly, and that calcium hardens plant tissue against the cold. He believed that.
Yep. Got another question here. In southeast Queensland, is 70% shade cloth perfect in summer, or would you recommend adding an extra layer of lower percentage on top of the 70? I've got aeroids in a growing greenhouse structure, and it's equivalent to 70% in there, and it's fine. I've also got some other things outside in a 50% shade house, and they just get left under 50. I think it depends on what you're growing too.
I grow everything under 50% except for the anthuriums, which I like to have them under 70 or even darker. And
I guess also knowing what you're actually growing and how it grows in nature is really important as well. There are plenty of anthuriums that do grow out in the sun, different species. Certainly a lot of the Pachynurum bird's nest style grow outside in the sun in very arid regions. So finding out a little bit about where plants occur in nature is going to lead you to make better decisions for that. This is a question, Steve, that I've got no frame of reference for, so I hope you've got an answer for it. Does anyone know if when crossing Thermatophyllum with Philodendron, should you include some Thermatophyllum pollen with the Philodendron pollen to place back on the Thermatophyllum flower, i.e. make a mixture, or is it best to use pure Philodendron pollen?
Look, you don't have to make a mixture. I wouldn't make a mixture. You want to know what you're going to get, don't you? You know, and
pretty much.
Be able to put a name on it. If you've mixed up pollen, you can't put a name on it, can you?
You're not going to guarantee, what?
Yeah, no, you'd use the same. Unless you want to make a hybrid, you want to cross a philodendron with a Thermatophyllum and create a hybrid, that's fine, nothing wrong with that, but you wouldn't be mixing up pollen.
No. I think it's just to use some Thermatophyllum pollen with the Philodendron pollen to break some pollination barriers,
perhaps.
Well,
one of the ways of doing that is to take some of the pollen and microwave it, and mix the microwave pollen with the unmicrowave pollen, and that tricks the female flowers into taking up the pollen. And that's a trick that doesn't only work with aroids, it's very useful with other plants.
So that'd be really good if you're struggling to sell a plant with its own pollen.
Yeah. Yes.
How long would you put it in the microwave for?
Only about 5 to 10 seconds, 30 seconds.
Not as long as in the blender.
No. Yeah, not long. Doesn't take long. Yeah.
Alrighty, we've got some plant samples that people have brought in, very kindly, and we've got a philodendron leaf that's. I think it's really just showing a little bacterial issue, and Steve and I are sort of thinking that it's probably more brought on from the cold weather. And
Too wet and too cold, I reckon, and the combination of wet and cold, which does cause problems. And it's easy to. There is a method of telling when you get a leaf malfunction and you suspect it's a disease. If you want to know test whether it's a bacteria or a fungus. The trick is called the bacterial ooze test, because fungi when it's in the leaf, it's in hyphae, long, thin, tiny threads, all matted together. Bacteria is individual cells. So what you do is you cut that leaf through where you've got the infected area, and you suspend it in a glass of water. Now, if it's bacteria, because bacteria is of individual cells, that the water around that lesion will discolour because the bacteria will come out of the tissue, and we call it bacterial ooze. And to use a callous term, it's just like pus from a boil. That's what, because a boil is bacteria. But if it's fungi, you will get no discoloration of the water around the tissue you've cut. Who knew that?
I did, but it was a long time ago.
You're not that old.
I probably wasn't listening in the lecture.
Nodding off, were we?
Yeah, well, it's after lunch. And this, we've got a sort of discoloured leaf with some yellowish spots, but really I think this is just, you know, more cold weather on a tropical plant by the look of it.
That's a typical cold weather effect on philodendrons. When you get that discoloration between the veins, see the brownish, yeah, that's typical cold weather symptoms.
So hopefully things will warm up in the next few weeks.
Yeah.
Steve, do you want to do that?
Yeah, okay. Andrew just asked me to show you how I do hemiepiphytic philodendron cuttings. So this is a redwings. It's really not rocket science. What I would do with this one, you can take single node cuttings, but I would take this as a cutting there, like so. Remove the leaf sheath, like so. Try not to damage these roots, and I'll take that leaf off, and that would be my cutting. Be careful not to get the sap on your hands, because some people can get irritated by it. So that's a really good tip cutting, but then you can also do the stem material. When I do the stem material, just take all of the foliage off. And the thing is, after you do this, after you make your cuttings, let them dry, let them callus. While that sap's weeping out, you should not plant them. So I'll take that internode space off. Now I'm going to lay these down. I don't leave any of these roots, then they won't be functional. The roots that are functional, and unlike me, use sharp secateurs. So you can get these roots cleanly off of the stem material, because they're non-functional now. The root that will be functional is the one that grows from the new leaf that comes out of the node. So get rid of all this. Get rid of all these roots. Now I can do 2 or 3 node cuttings, but for this I would go 2. So I'll take that out there, give me a cutting like that. Take that bit of internode away because it can often start to rot, and I'll get 2 more cuttings out of this piece, like so. Alright, so I've got 3 cuttings like that. Let them dry. Sometimes I'll so you can see there's the sap coming out already. Sometimes I'll cut them in the morning and then plant them in later on in the day in the afternoon, or sometimes I'll do them in the afternoon and plant them the next morning. And what I will use is just my regular potting media in a tray, and I just push them into the surface, so they're just sitting just about only about 10% of that stem material is actually in the media. It's basically sitting on top of the media, and don't give them a lot of water until they start to grow, and you will get a shoot, at least one shoot. Sometimes you get a shoot out of each node. When you're doing thicker material, you need it to dry longer. So you'd all be familiar with Philodendron Warskewixii, which can get like this thick. I would do the cuttings of that today, and I'll give them a full 24 hours before I plant them, so as they totally callus up. So, yeah.
Why wouldn't you go 1 cutting per node?
Oh, you can do single node cuttings, and if you have a very limited amount of material, you would do that. But I've got stacks of material of a lot of these things, so you get a bigger, stronger plant if you've got more reserve tissue there. And your chances of getting a growth are higher too, out of that cutting. All right? Some people are happy to do single node cuttings, and that's what people do with some of the rarer, more expensive stuff. I don't even do it with the rare, more expensive stuff. I do like to have 2 or 3 nodes on a cutting. If they're very close together, I'll do 3 nodes.
Is it worth leaving that internode section because that might have energy stored in it?
No, because the node acts as a barrier, as a temporary barrier, but if you leave a long section of internode space, it will start to rot. The closer you are to the node with your cut, the easier it is for the plant to callus and seal that wound, so as bacteria and fungal organisms can't get in. And the other thing is, some of them will have quite long internode spaces, then they take up a lot more space in your trays. All right, so when I plant these in shallow trays, not a bit deeper than that, but I plant these in shallow trays, and you don't want them to get them too wet. So I usually put them under a bench where they just get a little bit of a drip through from the plants that are on top of the bench.
What about the idea of painting with a fungal paint on each end to seal
Not recommended, no. Not recommended in this case because the healing process requires the presence of air. You know, you still see it for sale. I'm going off away from aeroids a little bit here briefly, but you still see it for sale in nurseries and shops, grafting mastic, tree wound and grafting mastic. It's been known for a long, long time, when you cut a branch of a tree, if you put that mastic over it, it seals it off from the air and it doesn't heal properly. It will heal faster if you leave the wound exposed to the air, because just like us, when we get a cut, it's the oxygen that makes our blood clot. Oxygen makes the sap clot. So that's why you don't put a sealant on them.
Question?
With the tip cutting, how deep do you plant that when you pop it in the pot?
Oh, yeah, well, I will just cover those roots to about there, where my feet... Yeah.
Alrighty. Well, one last one.
I was just curious as to if there was a reason why you left the roots on the tip cutting first, not.
Yeah, well, these are functional. These will pick up moisture and send it through to here. But when you haven't got that tip growth, these roots here will not continue to function. What makes roots take in moisture is the loss of moisture from the leaves, the transpiration process. All right, so it can't occur, and so those roots are no longer functional. And how are you going to plant that in a tray neatly with all that mass of root? And it's non-functional roots now anyway. It won't do anything. It'll just rot off, in fact.
All right. Well, I think if we...
You've got that question from Jenny too about the macrolobium.
Oh, question. Oh, there's part of a macrolobium here somewhere, yeah. Yeah, I did see it
Okay.
before. It was a, he's got it in there.
Pal's got it. Yep.
Okay. So the leaf of the Anthurium macrolobium has become yellow and blotchy, or chlorotic is the right word when the plant goes yellow. It looks like it could have spider mites. But it's hard to say without having a live specimen. It hasn't got spider mites?
It's white.
It's white. Okay, it's like an albino. Is it all over, the whole plant?
Pal, can we have a look?
Where? How are you showing?
I don't know because it's in with other
Yep.
stuff, and it's all close to the.
So it's in a warm spot, so it's not cold.
It's in a cabinet.
In a cabinet.
Not to grow lights, but other plants are in there.
It actually looks quite nice. If there's such a thing as a variegated macrolobium, that's got it. Yeah.
Well, that thing behind it's also damaged.
Yeah, and the other
just
thing is,
wonder-
I think it might be nutritional.
That's what I thought.
I think it might be nutritional because your veins are still green, your intervenal tissue is whitish. I suspect a magnesium deficiency. Okay, here's the test. Epsom salts. Give it a dose of Epsom salts and see if it colours up. Yeah, for sure. I reckon it's a magnesium deficiency because your veins are still green. When you get yellow or white in between green veins, it's usually 9 out of 10 times it's a magnesium deficiency.
Cool, thank you.
All right.
Steve, what's your answer for spider mite?
I get predators. I buy predatory mites from the company called Bugs for Bugs. I don't have much of a problem with spider mites, and you shouldn't have any at the moment. It's been a cold winter. They should all be dead, unless you've got a hot house where they can still keep warm and multiply in winter.
Very hard to get rid of.
Yeah, but that's why I use predators, because they develop resistance to the sprays that are on the market very quickly. Most of the sprays are very dangerous, most of them are very expensive, and they usually come in container sizes that are too big for me to ever use in the whole year. So I go for the predators, but just to a bit about the biology, ecology of spider mites. Generally speaking, a plant will attract spider mites to it, become susceptible to spider mite attack, if it's being grown in a condition where the humidity is considerably less than its natural environment. So the old trick years ago, take the parlour palm, Chamaedorea elegans, out of the South American jungle and use it as an indoor plant. Well, 9 out of 10 times they'll get spider mite because the air conditioning that people use or the heating they use dries the air out. So the plant that's grown in a very high humidity environment is now in a low humidity environment and it will get spider bites. So some people will say, oh, I don't have any trouble with spider mites in my shade house, but as soon as I bring a plant inside, I seem to get spider mite. And it's usually because the humidity dropped. Trick there is to put your plant in a saucer of water. So you maintain the humidity around that plant and you will reduce the incidence of it. Some people even use humidifiers in grow rooms, and one of the benefits of that, apart from providing a better environment for growth, it does reduce the incidence of spider mite. But the predators are not expensive and they're very effective.
So yeah. The predators then, Steve, how do you get them to establish? Because I've bought a heap of them too, and I've bought them at the periods of time to do a couple of inoculations with them.
Yeah.
So I just can't get them to establish.
Well, I've had very good results. I've had very good results from, but you need to target plants that are heavily infested when you first release them. I grow cordylines, and cordylines will be the first plants in my nursery to tell me if there's spider mites. And it doesn't happen every year. Maybe I only release the predators about once every 3 to 4 years.
We were getting them every 6 weeks from sort of July, August, September, when you get that drop in humidity, and it was producing cordylines commercially. And so, yeah, we were just keep pushing them into the system, and it took time, but yeah, we sort of got a level of control that we were happy with.
Yeah, and the thing about it is cordylines being such a common problem with them, and once you release your predators, if you move some of those cordylines around so they're in different locations, so you've got predators breeding up here, here, and over there because they've got plenty of food. If they run out of food, the predators die out. So I wouldn't be surprised if this summer I get an outbreak of spider mites because it's been so cold at my place that all the bugs are gone. There's virtually nothing attacking my plants at the moment as far as a mite or an insect.
I think they're a little bit more effective in a greenhouse situation to the predators. Like I've had predators in the gardens and same problem. They just disperse and you don't see them being outside.
Yeah, if they run out of food, they go looking for it, like I would. I put it into the potting media, into my orchid potting media. But if the orchid's been in the pot for some time, and quite often orchids are in a pot for a long time, I just put half a handful on top of the pot, spread it around. Takes a long time to go in, but it does go in.
Right, we'll finish up there.
Finish up there?
Thanks everyone for your questions. Thanks.